why are most cells small? So why did the designer gave so much importance with the sizes, okay, of these living entities? So we will try to answer that one with this statement, okay? So most cells are small and can only be seen by the microscope. So in the laboratory, you were being taught of how to use the compound light microscope or even the electron microscope. So there are some exceptions of these cells, like eggs of the most vertebrates, fishes, amphibians, reptiles, and birds have and some of the long nerve cells has a slightly bigger in size, and it can be seen by the naked eye. So one reason for the small sizes of your cells is that the ratio of the volume of the cell's nucleus to the volume of its cytoplasm must be so small that the nucleus or the major control center of your cell cannot control the cytoplasm. Okay, so another aspect of your cell volume is working with its limited cell size. As the radius of your cell lengthens, cell volume increases more rapidly than the surface area. So we are much concerned with the size of your cell, okay, or the volume of your cell, which is increasing, okay, than the surface area, because it holds different organelles. It must have to be a larger volume, because organelles are working, okay, for the nourishment of your cell. Like different metabolic functions are also portrayed in your cell. So this is just a figure, shows that the radius, the surface area, and the volume of your cell. So try to look at this surface area. We have 50.26 centimeters squared, and the volume is slightly smaller. When you try to increase surface area, then the volume is also increased in centimeter cube. So we will try to divide the surface area over the volume. So we got here the numerical data, okay, so it's 0.75 here, 1.5 and 3. So the cell membranes are, again, earlier I discussed about the importance of these plasma membrane, because they regulate and perform cell communication. It guards the exit and entry of certain molecules, certain solutes and solvents. So it surrounds your cell, okay. Other membranes inside the cell has to enclose the organelles and have properties which is similar to your plasma membrane. So we have Jonathan Singer and Garth Nicholson, which developed the fluid mosaic model of your membrane structure. And according to this model, a membrane is a double layer or a bilayer of proteins and phospholipids, and it is fluid rather than solid. So a phospholipid bilayer is forming a fluid C, in which a specific proteins will bind to it like icebergs. So just imagine how it works. And this is a fluid mosaic model of your membrane structure. So it's wavy because it contains the bilayer, the phospholipid tail, okay, and the phospholipid heads. So they have cholesterol molecules sandwiched in between. So this is the outside of your cell, and this is the cytoplasmic side of your cell. Embedded inside are the fibrous proteins, which passes through the membrane, and a globular protein, plus the existence of carbohydrates, okay, the macromolecules found outside of your cell membrane. So we'll discuss why does a glycolipid found outside, not inside, and so on. So the fluid mosaic model of your cell membrane has in a constant state of flux, shifting and changing while retaining its uniform structure. So the term mosaic refers to the different kinds of proteins dispersed in your phospholipid bilayer. Okay, so one of the most important points of your fluid mosaic model, so the phospholipids have a polar end and one non-polar end. So these polar ends are oriented on one side towards the outside of your cell and into the fluid of cytoplasm on the other side, and the non-polar ends face each other in the middle of the bilayer. So we have the tails, okay, of your cell membrane that are hydrophobic. They don't like water. They are water dreaders, and as a result, the polar spherical heads, okay, or the phosphate portion, are hydrophilic. We call them water attracting molecules. Also, with cholesterol is an important feature of your plasma membrane and organelle membranes of your eukaryotic cell. So it's embedded in the middle of your cell membrane to make the membrane less permeable to water-soluble substances. Okay, and it makes the rigid structure of your cell membrane. And the membrane proteins are, or the big chunks of proteins which is attached in the inner or outer membrane surface are embedded on it. We call it peripheral and intrinsic